Hello, and welcome to our presentation, Can CRISPR Correct Cancer?, specifically on genome editing and immunotherapy. We'll be discussing how CRISPR, a gene editing breakthrough, could transform the way we diagnose and treat cancer, focusing on both the science and the broader ethical and clinical picture. Here's what we'll cover in this talk. First, I'll introduce what CRISPR is and how it works. Then, we'll talk about some cancer and DNA mutations, how CRISPR fixes cancer genes, CRISPR in cancer immunotherapy, clinical trials and results, pros and cons, the ethics and societal impact of CRISPR, and then some future directions that this could go to. Before we start, I just want to enunciate that the specific area of CRISPR we'll be looking at is not CRISPR as a whole with every organ, but very specifically CRISPR with cancers and tumors. So, what is CRISPR? CRISPR, which stands for Clustered Regularly Interspaced Short Palindromic Repeats, is a revolutionary genome editing tool originally discovered as part of the bacterial immune system. Scientists have adapted this system for use in humans by combining CRISPR sequences with a specialized enzyme called CAS9. CAS9 acts like a molecular scalpel. It can be guided by a short RNA sequence to almost any location in the genome to make a precise cut in the DNA. This enables researchers to delete, modify, or replace single genes, making CRISPR far more efficient and flexible than earlier gene editing technologies like zinc finger nucleases or talons. Today, CRISPR is used across biomedical research and is making its way into clinical medicine, especially for genetic diseases and cancer. So, before we answer the question of cancer and DNA mutations, we have to go into cancer at its core. And cancer, at its most fundamental level, is a disease of faulty DNA. Mutations accumulate over a person's lifetime, are sometimes inherited, and can either activate oncogenes like KRAS and EGFR. KRAS is a genetic mutation that occurs in the 12th amino acid, or disable tumor suppressor genes like TP53. These driver mutations disrupt the normal checks and balances on cell growth, allowing uncontrolled proliferation that leads to tumors. Traditional therapies like chemotherapy, radiation, and surgery aim to kill or remove rapidly dividing cells, but they often don't address the underlying genetic cause. Targeted therapies, and especially gene editing by CRISPR, offer the promise to correct these mutations at their source, potentially providing more durable or even curative results. In both lab settings and early human studies, CRISPR has been used to repair or knock out cancer-causing mutations directly in DNA. One approach involves taking blood or immune systems from a cancer patient, editing their DNA outside the body to inactivate certain genes, and then returning the corrected cells to the patient. This method is called ex vivo editing, and it has been the most effective for blood cancers such as leukemia and lymphoma, because blood cells are relatively easy to remove, edit, and reinfuse. Early trials targeting TP53, KRAS, and other genes show that editing is possible but not yet efficient or comprehensive for all tumor types. A rapidly growing application for CRISPR is in boosting the power of immunotherapy, which is treatment that helps the patient's immune system attack cancer cells. CRISPR allows scientists to engineer T cells, one of the immune system's main weapons, so that they express synthetic receptors or eliminate the genes that inhibit immune attack. This dual approach means that T cells can now better recognize tumors and ignore the cancer's usual defense tricks, making them more effective and longer lasting inside the patient. These advances aren't just theoretical. CRISPR enhanced CAR T and edited T cell therapies are now in clinical trials, showing early evidence of increased anti tumor activity, fewer side effects, and improved patient outcomes. Currently, there are more than 150 gene editing clinical trials underway worldwide, and dozens of these are using CRISPR technology for cancer. Other trials are tackling difficult cancers such as glioblastoma by using CRISPR to disrupt certain genes or treating metastatic solid tumors with T cells engineered to lack the PD-1 gene. Most of these early phase trials are currently showing that CRISPR edited cells, can persist and function in the human body, with mostly mild side effects, although durable remissions remain uncommon and additional optimization is required. Major technical obstacles include achieving full delivery to tumor sites, refining editing precision, and ensuring long-term safety. In terms of pros of CRISPR, CRISPR enables highly specific corrections or deletions of the genetic mutations that actually drive cancer. Therapies can be customized down to the individual patient's tumor genetics, potentially leading to more durable remissions and fewer side effects. This technology has the potential to transform both cancer treatment and prevention, especially for those with inherited cancer risks. With cons, delivering CRISPR accurately to all tumor cells, especially in solid tumors, remains a significant and unsolved problem. And right now, treatments are expensive and likely to be offered only in specialized clinics, limiting access for many patients. Of course, gene editing for cancer, as all biological studies, raises complex ethical questions. Will high costs put these therapies out of reach for most cancer patients, increasing health disparities between the wealthy and uninsured or underinsured, especially with all we have going on in the world of healthcare subsidies? Another concern is the potential for germline editing, deliberately making changes that can be inherited by future generations, which risks rekindling eugenic ideologies and raises questions about consent and social justice. Strong, globally coordinated regulation, as well as inclusive public debate, will be essential to ensure CRISPR is used for responsible medical progress and not for unethical enhancement or discrimination. Looking ahead, the goal is to make CRISPR both safer and more broadly accessible, refining accuracy, minimizing off target risks, and combining gene editing with other established cancer therapies. Long term, CRISPR could revolutionize cancer care worldwide, but success will depend as much on ethical decision making and access as on scientific innovation. All claims and results in this talk are grounded in peer reviewed studies and leading clinical trials. And thank you guys for listening. If you have questions, please refer to the references or ask me or Lizzie for more detail. Thank you.